We are interested in the discovery and development of agents for the prevention of cancer. Primarily, or historically in our lab, this has been dietary derived agents, but we're also now looking at drugs that have been used for other purposes and for other indications and trying to use those in cancer prevention as well. Most of our work has been on compounds such as resveratrol, which is in red wine and red grapes and peanuts, and curcumin, which is in the spiced turmeric. And we've been looking at those primarily in the prevention of colorectal cancer, so bowel cancer. We have to be very careful how we talk about our results because it can very easily be taken out of context. Particularly, we've done a lot of work recently looking at resveratrol, so the compound in wine, and trying to compare the activity of a dose that you can get through the diet and a dose that we've used in clinical trials before. So the dose you get through the diet, I tend to describe it as the amount you could get in a couple of large glasses of red wine. Obviously, there's no wine there in what we do, but I have to be very careful because then come the headlines. Yeah, wine prevents cancer, and it doesn't, but the compound in it can prevent cancer in our laboratory models. For the resveratrol work, we use a synthetic version of resveratrol, so it's not an extract, although some other people probably use extracts. For the curcumin work, we use an extract of the turmeric. So that's a bit different. We have done some work on plant extracts and berry extracts where you can look at compounds called anthocyanins. But it's, and that has its advantages as well. But obviously, you're looking at a mixture. You don't know what's causing the effect. So I quite like to make it more basic, make it more simple, and you know what compound you're looking at, and you can try and figure out what mechanisms it's working through. And then optimise the doses to take in the clinical trials rather than having a mixture where you've just got too many unknowns, I think, at the moment. We've done a bit in the past on prostate cancer. We have done some trials in prostate cancer patients, and we are beginning some work on lung cancer, but primarily it's been colorectal. It's a really good model. What we always have to keep in mind is that there is a clinical model in which we can test our agents in clinical trials. There's no point doing loads of lab work if you can't then take that into a patient group. And colorectal cancer is a really good model because you can identify patients that are at high risk because through the bowel cancer screening program, so patients that have adenomas detected, they have them taken away and then they're at increased risk of adenoma recurrence or colorectal cancer development. So there's quite a nice group of patients that we can intervene and give our agents to and see whether they have effects on those patients. We do a lot of phase 1 trials trying to see how much of the compound actually gets into the target tissue, how much gets into the blood, because that's a big problem with this kind of area of research, particularly with dietary derived agents, but also with drugs. There's a lot of papers in the literature where people use concentrations that are 100-fold higher than what you can ever actually get in a person. So we find it's really important to do those early phase trials, get the pharmacokinetic data and feed that back into our preclinical work. So we make our animal models and our cell culture work as representative of the clinic as possible. So we've done a couple of studies looking at resveratrol safety and pharmacokinetics, and in one of them we used a radio-labelled form of resveratrol, so C14 labelled, gave that to patients that were having surgery for colorectal cancer, and we gave it to them at 2 different doses. So here we were comparing the dietary dose with the dose 200 times higher, and we just wanted to see whether it got to the target tissue, how much got there, and what was the metabolism like. And we found that at both doses, so the low dose and the high dose, you do get resveratrol in the colon. And we also did a study in prostate cancer patients and found that in the prostate tissue, the profile is very different. So you have mainly metabolites in the prostate and not really any detectable resveratrol. So that's going to have an impact on whether resveratrol might actually work in a person for the prevention of prostate cancer. And then taking those results, knowing that the tiny dose of resveratrol can get to the colorectum, we did studies using the APC min mouse, which is a mouse model where the animals spontaneously develop colorectal cancer, or polyps in their intestine, and we found that the low dose is more effective than the high dose at preventing preventing intestinal adenomas in these mice. So that's suggestive of maybe at least a non-linear dose response, or maybe even a bell-shaped curve or a U-shaped curve with a a bigger effect at low doses. So the patients was purely a phase 1 trial, so there wasn't any follow-up there, but it is our intention for both resveratrol and curcumin to do a trial in that adenoma setting that I mentioned. So we would aim to recruit patients and then give them resveratrol or curcumin or aspirin for a year, and then look for evidence of activity in the blood of those patients over time to see whether the doses we've chosen are potentially active in those patients. So that's planned for the next couple of years. We were kind of using it as a positive control in this trial because, yeah, there's excellent evidence that aspirin does work. But what we don't know is we need ways of monitoring whether it's working in individuals. So the aim of including aspirin here was to try and identify biomarkers in the blood that we can detect, we can analyse proteins in patients, hopefully, to say, yes, it's working in you, it's not working in you, you should continue, you shouldn't. And using it as a way also to optimise the dose of aspirin, because that's not quite clear yet. I mean, we know that the lower doses and the higher doses can have activity in patients, in people, but it's not really nailed down what is the best dose for people to take. So we might be able to use these biomarkers to help with that decision as well. Based on what I've heard, say, over the last 6 months, definitely. I think when I hit 45 or so, I'll start taking aspirin. I know a lot of people that do take resveratrol and even curcumin. We often get phone calls and emails from patients that want to take these agents, but at the moment the evidence isn't there, so I wouldn't. No, not at the moment. But aspirin, the evidence is there, so I definitely consider it when I get a few years older.